My name is Bernal, and my presentation was over exploring the pitfalls and possibilities of gene therapies. So I wanted to begin by going over congenital disorders, giving a little background. Congenital disorders are conditions that are existing at birth, and they can be caused from a variety of things, from non-functioning or missing genes to mutations induced on the DNA, or full or partial extra copies of chromosomes being present. And the side effects of these diseases can also range anywhere from developmental delays to even death, just depending on the severity. And I'd also like to note that the disease typically affects each individual a bit differently. So to correct these type of disorders, that's where geneticists have come up with the idea of gene therapy. The most typical way that this is done is by transplanting normal genes into cells to compensate for defective or non-functioning genes. This type of research brings along some cultural, religious, and ethical concerns. And over this presentation, I will cover the types of cells affected as well as the challenges faced. So, thousands of Americans are born with genetic disorders annually, according to the U.S. Department of Health and Human Services. And as I previously mentioned, these diseases can affect the individual in a wide array of symptoms, ranging from mental and developmental delays to impairing motor function to even causing death, in some cases hours, if not days after the baby is born. And this figure here depicts an abnormal blood cell compared to a normal blood cell in sickle cell anemia. You can see here that it is misshapen. And this disorder here is caused from a faulty gene as well that's responsible for the formation of red blood cells. So I just wanted to give an example there. So the types of cells that are involved are somatic and germline cells. These are the ones that are typically targeted. And I'll start with the less controversial of the two, somatic cells. Now, somatic means pertaining to the body. So when these types of cells are targeted, only the individual is affected because these cells do not reproduce. And a type of way I would consider this is kind of chemotherapy. It affects just the individual. Germline therapies, on the other hand, involve gamete cells, like ova and sperm. So this is a more controversial method because alterations to the genome are passed down to offspring. So this can inherently cause some effects to the familial lineage. So both of these therapies aim to correct non-functioning genes or alter gene expression in some sort for potential disease eradication, whether it be in the individual or the population of the family. So challenges and concerns. Do humans have the right to alter genomes? This brings about some ethical and religious beliefs as well as some cultural concerns. Those are more religious believe that a higher power is the only one who has the power to make such alterations. Other critics say that too little is known right now to make these permanent changes and it may not be safe. And long-term side effects are not known as of right now because the studies are in such infancy still. So germline mutations can cause irreversible effects in offspring that cannot be changed back, as well as you can't really set up proper consent for the individual when you're altering the DNA before the individual has a chance to have a say in it. And all of these challenges make funding a challenge within itself. So our proposed solution and possibilities is really the only thing that we have is to conduct more research in order to get the answers. So this would be beneficial for those to offer relief that are living with diseases such as sickle cell anemia. Imagine a day where this disease is eradicated completely, and that is the ultimate goal, to just get rid of these disorders that are typically inherited, whether it be a recessive or dominant gene. So real quick, I'll just brief this because it would take up the whole presentation, but my study findings were that Jacobson and about 8 others wanted to kind of investigate this. It's called a gene RPE65. So what you have here is a normal topographic view of the retina compared to those individuals who have mutations in this gene. And you can see the differences there. So their work concluded that large and small animal models have been treated successfully with gene transfer. And so I believe that gene therapies are worth pursuing in human trials because they have the potential to eliminate the onset of disorders and also to offer hope to individuals that are living with congenital diseases. I'm sure it's something difficult and it does hinder their way of life. So this would be something just groundbreaking for these individuals. As well, I know we have to address the concerns. I feel as though if consent is carried out properly to ensure protection for those involved, there's more good than bad. And human trials will help us to gain so much insight when studying these particular diseases in the hopes of perhaps finding solutions for other diseases that stem from these. And those were my references. Thank you.
All right.